As the world is inching ever closer to eradicating the polio disease, an unlikely candidate, one of two types of vaccinations against the polio virus, is posing a threat to eradication. In the following video, we will tell you about poliomyelitis, the polio virus, the currently available vaccines, and their relationship to the last remaining cases of poliomyelitis in humans.
We hope to not only inform of the details of the virus and the varying vaccines, but also to argue the importance of not using the anecdotal evidence of a particular polio vaccine to push an anti-vaccine agenda.
Poliomyelitis, most commonly known as polio, is a viral disease which targets the central nervous system. Polio is caused by the poliovirus, which is a positive-sense single-strand RNA virus composed of an RNA genome inside of a protein capsid.
As a positive-sense virus, unlike DNA or a negative-sense RNA, it directly targets translation in the human cell.
The virus enters human cells by binding to the CD155 receptor, also known as the PVR or poliovirus receptor. When the viral particle enters into the cell, the viral RNA is released. The RNA genome acts as a messenger RNA which commands the cell's translation machinery. This stops protein synthesis and initiates the synthesis of viral proteins.
The poliovirus is typically spread through the ingestion of infected fecal matter through the mouth, but can also be spread through the ingestion of other substances such as food or water that have been contaminated with infected fecal matter.
Typically, the poliovirus has no signs of infection. However, some infected individuals display a range of symptoms, including a sore throat, fever, headache, stomach pain, and other flu-like symptoms. A small portion of people infected with the virus will show symptoms that affect the brain and spinal cord, such as paralysis, paresthesia, and meningitis.
Varying treatments can potentially help, but once infected, there is currently no cure. That being said, there are two types of vaccines that will prevent infection of poliomyelitis. These are the inactivated polio vaccine, or IPV, and the oral polio vaccine, or OPV.
The IPV vaccine was invented by Jonas Salk and first came into use in 1955. Since 2000, IPV is the only polio vaccine that has been administered in the United States. The vaccine is produced from wild-type poliovirus, which has been inactivated or killed with formalin and contains inactivated strains of each serotype of the poliovirus. The vaccine is administered in a series of 3 shots, but it is typical for a 4th booster shot to be administered as well.
The OPV was invented by Albert Sabin and was first used in 1961. There are variant types of OPV which contain 1, a combination of 2, or all 3 serotypes of the poliovirus.
OPVs have been the most common vaccine used for a number of reasons. First, they are cheaper than the IPV, and they are administered orally and therefore do not require medical professionals or sterile syringes. Furthermore, OPV immunization can lead to accidental immunization of others in areas with bad sanitation or hygiene. This can occur because the vaccine virus replicates in the intestine for several weeks after administration.
However, unlike the IPV, which contains an inactivated poliovirus, the OPV contains a live virus. Although extremely rare, this live virus can mutate and reestablish neurovirulence. According to the Polio Eradication Initiative, in 2015, there were 96 identified cases of polio across the world.
70 of which were caused by the wild strain of the virus, 26 of which were derived from the orally administered vaccine. Although 26 cases globally is statistically low, the Polio Eradication Initiative has identified that the OPV poses a threat to global eradication.
As a result, in 2014, the World Health Organization, through the targeted efforts of the Polio Eradication Initiative, developed a plan to phase out use of the OPV by 2020.
It is important to understand the aforementioned differences in the biology of the OP and IP vaccinations in order to prevent utilization of the dangers of OPV as anecdotal evidence in an anti-vaccine agenda. While incredibly unlikely, the OPV possesses the potential danger due to the live virus that the vaccine contains. This means that on rare occasion, the live virus would be able to mutate and infect the vaccinated. The IPV does not contain a live virus and therefore does not pose the same threats as the OPV.
Despite the very low rate of mutations occurring in the OPV, the World Health Organization has developed a transition plan to phase out OPV in order to attain worldwide eradication of polio.